The next contestant is Jiayi Peng. She takes us to the world of physics. She comes to us from Horace Greeley High School in Chappaqua, New York.
Good afternoon. My name is Jiayi Peng, and today I'll be talking about my research project on modeling the neuronal network. Neuroscientists today are starting to unravel the brain's complexity by studying its collective dynamics as opposed to the individual neurons. I'll start off with some basic information about the neuron network. Neurons communicate by receiving signals from surrounding neurons through connections called synapses. Once the accumulation of these inputs reaches a certain threshold, the neuron fires an action potential and redistributes the signal to surrounding neurons. This may cause others to fire, leading to a cascade of firings called a neuronal avalanche. In 2003, Dr. Beggs and Dr. Plenz were the first to find the parallel distribution in neuronal avalanche size. This suggests that the neuron network operates at the critical point, since the parallel distribution is the signature of the critical point. So why are scientists interested in criticality in the brain? Well, several information processing functions are optimized at the critical point, such as dynamic range, computational power, information transmission, and information capacity. As you can see on the right, dynamic range is optimized at the critical point. The critical point is an optimum state poised between two extreme states, an activity dampened state called the subcritical state and an activity excited state called the supercritical state. In the subcritical state, information is lost from the green inputs to the red output, and in the supercritical state, information is exaggerated. Scientists have also found that deviations from the critical point can be linked to devastating neurological disorders. For example, the subcritical state has been found to be linked to Alzheimer's and autism, while supercritical state is linked to Parkinson's disease and epilepsy. There are two types of criticality in physics. One is parameter-tuned, which means that the system arrives at criticality through tuning a control parameter. For example, in the ISIG model, temperature is tuned. The other type is called self-organized criticality, or SOC. The simplest form of SOC is the sand pile model, which models avalanches in a pile of sand. Criticality in the neuron network is believed to be SOC. Several models have been proposed to explain how synaptic plasticity, or the ability for synapses to change strength, could cause self-organization. Two types of synaptic plasticity, short-term and long-term synaptic plasticity, have been modeled separately in previous work, such as the LHG and the Tesloff et al. Models. Thus, it is known that both timescales of synaptic plasticity can individually drive the network to the critical state, but it is also known that they are both present in real neuronal networks. So the question is, how could these two timescales of plasticity work together to produce SOC? Would it be possible to combine them both into one model and partition their effects? I proposed a new model that combines short-term synaptic plasticity with long-term metaplasticity to investigate their effects on self-organized criticality. First, I tested to see if my model could produce critical results from various rigorous tests in the field of critical phenomena, such as the ones above. Then I tested to see if that criticality could be maintained in the case when it's perturbed. My model combines the simplicity of the cellular automaton method of the sand pile model with the neuronal features such as short-term and long-term synaptic plasticity. The benefits of such a model include being able to study large system sizes that was not possible in the LHG model, to model different topological structures, and to partition the effects of the two timescales of plasticity. Here are two diagrams that help explain the dynamics in my model. First, I studied a square lattice. Here, the neurons are represented by circles, and the synapses are represented by the arrows. Both the neurons and the synapses have individual dynamics that increase and decrease their strengths. I also studied multiple randomly connected networks, such as the one below, and a fully connected network. Here's a simple flow chart of the code in my model. First, the system is driven, meaning one random neuron is stimulated. If its membrane potential passes the threshold of 1, it fires an action potential and redistributes the signal to surrounding neurons. Once the synapses are used, they undergo synaptic depression, which decreases their values, and all synapses undergo spontaneous recovery, which increases their values. These two features make up the short-term synaptic plasticity. The model then checks if there are any more membrane potentials above the threshold of 1 that would fire. If there are, the process repeats. If there aren't, the avalanche is over and long-term metaplasticity kicks in to change the synaptic depression rate U. I will first study the system without metaplasticity and then with to partition the effects of the two timescales of plasticity. I will later be talking about avalanche size distributions and temporal shape scaling. To understand avalanche size, here's an example avalanche where the red neuron was the first to fire. The activity then propagates the purple, the blue, the green, and the yellow neurons during time steps 2 through 5, respectively. The size of this avalanche is 11, since 11 neurons fired, and the duration is 5, since it lasted 5 time steps. On the right is an example of an avalanche shape, which plots the number of firings against their respective time steps. To see if the results produced by my model coincide with the experimental results, I studied the size distributions of the 3 states: subcritical, supercritical, and critical. The turquoise curve is the size distribution of the subcritical state, which is characterized by a quick cutoff showing that there are not many large avalanches. The size distribution of the supercritical state is characterized by an arc indicating that there are many large avalanches, and the size distribution of the critical state has the longest parallel range, which is cut off by the effect of the finite system size. Even though the parallel is the signature of the critical state, alone it does not validate that the system is operating at the critical point. According to the theory of critical phenomena, there are 2 types of lengths that can cut off the parallel range. One is the system size, whose effect is shown, and the other is the correlation length. I will show that these size distributions can be scaled by both of these lengths to confirm that the results come from a critical process. I was able to scale the size distributions of different system sizes you just saw onto one curve through a process called finite size scaling. As I just mentioned, similar to the effect of system size, the correlation length related to the synaptic depression rate U can also cut off the parallel range shown here. Here, I was able to scale the size distributions from the previous slide onto one curve through a process called scaling of non-critical states. Both finite size scaling and scaling of non-critical states confirm that the system produced results from a critical process. Furthermore, I studied the internal structure of the avalanches by looking at the avalanche shapes, which can be scaled by the durations ranging from 5 to 20, shown here. This process of avalanche shape collapse is a new, more stringent test to verify that the system is at criticality. I also looked at the relationship between the 2 parameters, the critical value UC and the system size N. To study the effect of different topological structures, I studied the relationship between the critical value UC and the number of neurons each neuron is connected to, NN. Since short-term synaptic plasticity can attain criticality, I tested to see if it could maintain it in the face of perturbation. First, I let the system run at criticality and then perturbed the synaptic depression rate U to a non-critical value to see if the system could return to criticality. I saw that neither perturbation to the subcritical state in light green nor perturbation to the supercritical state in light blue could return to criticality. The dark green, the dark blue, and the red size distributions correspond to the unperturbed original size distributions shown on my first results slide. Since short-term synaptic plasticity is not sufficient to maintain criticality, I tested to see if the addition of long-term metaplasticity could help the system recover from the perturbations. Sornet states that one way to achieve SOC is to have a nonlinear feedback of the order parameter onto the control parameter. I implemented this idea with the above equation to add long-term metaplasticity to my model. With the addition of long-term metaplasticity, I saw that perturbations to the subcritical state, green, and perturbation to the supercritical state, blue, could return to criticality since their size distributions are almost identical to the critical parallel in red. To more closely look at the effect of long-term metaplasticity, I graphed the synaptic depression rate U over time after perturbing it. As you can see, the U converges over time to a critical value, indicating that the system is self-organizing. In conclusion, I built a new cellular automaton model that combines short-term synaptic plasticity with long-term metaplasticity to investigate their effects on attaining and maintaining operation at the critical point. I examined the criticality in my system through size distributions, finite size scaling, scaling of non-critical states, and temporal shape scaling. I found that short-term synaptic plasticity is sufficient to attain criticality, but not to maintain it. But with the addition of long-term metaplasticity, the system can recover from perturbations that are prevalent in biological systems. Working together, these two timescales of synaptic plasticity can help a system both attain and maintain operation at the critical point, leading to a self-organized critical system. I'd like to acknowledge my mentor, Dr. Beggs from Indiana University, and my science research teachers, Dr. Gessler, Ms. D, and Mr. Goodman. I'd also like to thank the Siemens Foundation, the College Board, George Washington University, and the judges. Thank you all for your time. Here's my selected bibliography.